Yes, so thank you again for the invitation. And, I'm a little hesitant to speak now after two great NanoSIMS experts, that showed you very much the deep workings of the NanoSIMS as well and how you can get some really cool work done with it. We are not, like a disclaimer from my side, I'm not a NanoSIMS expert. I am like a dumb biologist that ended up using NanoSIMS at some point and kept using it because it's the only way to answer some questions. So I'll kind of like give you an idea about our ride into the field of eventually NanoSIMS. But the previous two speakers are far better experts. So for technical questions, I'm probably not the best, but I will try to answer if you have some questions. So let's move on. So we are synapse biologists. What we like to do is to look at synapses and see how the molecules there behave. You see here on the right hand side some simulations that interpret a lot of imaging data that we have about how things move in a synapse. On the left-hand side, you see the composition of the synapse in the brain, with all sorts of molecular arrangements and whatnot. I mean, this is what we like to look at. I mean, we go deeper and deeper using fluorescence, and this is from a recent bioarchive that we have. We now have fluorescence tools that, at least to in our opinion, go down to submolecular resolution. You can see here in fluorescence some IgGs, some IgAs, some IgM molecules. So we can do a lot of work with fluorescence, and it's nice and it's interesting. But there's one problem. This typically, the fluorescence shows you the here and now things. I mean, the fluorescence can show you stuff over some days, maybe about a week or so, but it can't typically show you things over, let's say, weeks and months and maybe the lifetime of an animal. Then you need other types of imaging approaches. And this is why we ended up using SIMS. So how can we investigate long-term changes? And let's start with one problem which we faced a few years ago. The problem, as an example, was erythropoietin. This is a doping reagent. You have possibly heard over the last decades, a lot of cyclists, bicyclists, they used erythropoietin to dope themselves to get better muscles, to get better heart. Long distance runners used it, and so on and so forth. It improves the blood flow and heart performance. And a very good friend of ours, a colleague here in the city of Göttingen, in the middle of Germany, she did the following experiment. She took mice, injected erythropoietin into these mice, and guess what happened? You have a task here, and essentially you're looking at the percentage of the mice that don't finish this maze task. The normal mice, take some time to do the task, to learn it and to do it well. If you inject erythropoietin in mice, they do the task in less time. It takes shorter. They get smarter. They can finish basically learning things faster. So you could explain this in many ways, but you could have many hypotheses. But what is actually going on? What is this weeks-long change in the brain of a mouse? Well, it wasn't easy to look into, and definitely with fluorescence, we just didn't have any clue how we could actually learn this. So what does it do? The easiest thing is maybe you get new neurons, and maybe this is also what happens to the humans that inject erythropoietin into themselves. So we would need to look over weeks and months into these mice. Well, how can you do that? One approach is, as you already heard from Dr. Steinhäuser, you already heard you can take mice, you can feed them with an isotopic food and like 13C lysine or something like that. Then you can take the brain and then smash it up, and then you can look into the lifetimes and changes of different proteins. That's for sure, you can do that. This is the classical SILAC approach. We do this as well. We can tell you how old all sorts of proteins are in the mouse brain, but you want to see it. So then you need a machine to image the isotopes, and that's the theme of today's seminar, of course, the NanoSIMS. So what you do is you take your beam, you're scanning the surface of the specimen, and you're exploding your secondary ions, and then you're measuring your secondary ions with a lot of detectors in this MEMS procedure, which was introduced in the first lecture already, and which is a stunningly nice procedure. You can really get a lot of information. Now, I'm a resolution person, sort of always obsessed about, how good is the resolution? Well, we never really got over about 50 nanometers or so. These are little gold particles, and you can measure them, run a scan over them. The actual measured resolution is somewhere around 50 nanometers in our hands. And if you go to let's say, a more like naturally occurring isotopes like nitrogen, 14 nitrogen in the form of the CN minus molecules, little ions, these give you an apparent resolution of about 100 nanometers, give or take, 15 N, also more or less in the same ballpark. So you get a decent resolution. What is your resolution in the z-axis? That's actually pretty good. You can just keep on taking images into a specimen and just see how much you're milling away with every section, which is give or take more or less your actual resolution of what you're getting out of the specimen. And if you look at this type of measurements, you get an idea of somewhere around 5 to 15 nanometers in plastic embedded classical samples. This is not too bad. I mean, in the z-axis, this is actually very good. In the XY, it's decent. It's certainly decent. It's on a par with some of the super resolution methods in optics, worse than others, but it's decent. This still can give you a pretty good picture. So how do you then know what each structure is? What is the nature of each structure? Sometimes the nature is obvious. You're looking here at a heart muscle, and you have 1, 2, 3 blood vessels in this heart muscle, and you have here 1, 2, 3, 4, 5 little cells in the muscle. This was a mouse fed with 13C for some weeks. And then if you look into these blood vessels, you see 1, 2, 3 cells here, but these 2 cells marked with the blue stars, they are not existing. So if you do some form of ratio, no, you don't see them. 2 are not really existing. The other 3 are quite bright. What's going on here? You're looking at erythrocytes. You're looking here at erythrocytes in the blood capillaries in the heart. And these 3 are brand new erythrocytes that have been made out of new protein. And these 2 here are old erythrocytes that have a lot of old protein. Very simple. Identity is pretty obvious. You can move on. Sometimes it's not so obvious, so you can correlate things with histological labels. So you can just do embedding in plastic. You can do some form of, let's say, toluidine blue or any other type of staining here. And you're staining your proteins in blue, and then you get a pretty good picture of what's going on in your sample. Not a superb resolution, but still a decent resolution. And then you can combine this with the NanoSIMS. You get an idea of what is where. If you want to get even better, you can try a correlation with fluorescence. So what you can do is you have your organelles here. They are immunostained for different markers. You have here TGN38, a marker of the Golgi apparatus, synaptophysin, which marks sort of like a precursor form of vesicles. Calnexin is the endoplasmic reticulum. You can do a super resolution, of course, if you want on this type of fluorescence, and then you can combine it with the NanoSIMS pictures. And these were neurons fed with nitrogen 15 containing amino acids, and you have a ratio of N15 to N14, which gives you sort of like a picture where you see this tongue-like thing and this circle-like thing and this shoulder here. All of that kind of overlaps quite nicely with the Golgi apparatus marker. And then you can kind of learn from such pictures that your Golgi apparatus is the place in the cell where brand new proteins are being modified and are being kept for some time. Turns out the endoplasmic reticulum, which is the first place where you make your membrane proteins, for example, that is less intensively occupied by new proteins. Then it's less than the Golgi. The Golgi, presumably, that's where they stay longer. So then the amount of new protein per pixel is higher in the Golgi apparatus than in the endoplasmic reticulum at any one time. This is in neuronal cells. It may not be the same in every other cell type. So this kind of works, the correlation with fluorescence. It's easy to do, very easy. you have your animals or your cells grown in presence of an isotopic metabolite. Then you do your immunostaining or any other type of labeling you may want, embed the samples in plastic resin, and just do whatever resin you like. I mean, there are many types that could work for this. We like LR white, LR gold type of resins because they are good for fluorescence and for SIMS and for electron microscopy, should you want to do that. Then you cut them with a microtome. We find that like 50 to 200 nanometers is best, most likely about 100 to 200, 220 nanometers, that works best. Easy to cut, 100 nanometer is so trivial to cut on an ultramicrotome, no problem at all. Then you can image these things in light microscopy. There's only one trick. You see the wafer here, it's made of silicone. Silicone is not transparent. So it's not as convenient to image these samples as on a normal, let's say, cover glass, because when you flip it and put it down, you don't actually see the light and you don't see very well where your objective is positioned because, well, it's not transparent. So it takes a little bit of getting used to really scan these things properly, but everybody in my lab who did this got it to work fairly well after some weeks. It's not that difficult. It's just a little bit of an annoyance, but you could use also cover glasses that are coated with a coat that makes them electrically conductive, and those cover glasses are still transparent. That would work as well. We just are lazy. We never really went to these cover glasses. We just were left with the silicone wafers. Then you image these things with light microscopy in any way you want. You can burn sort of markings into the plastic. You can take a high power laser and kind of like burn markings there to find exactly the cell. We did this in the first couple of projects, and then we realized it's useless. You don't need that. You can just take your original pictures and correlate them directly with the pictures in NanoSIMS. There's no problem in finding the shape of the same cells and finding the same objects. So, you know, this burning thing that we always used to do, we don't do it anymore. It's just not necessary. And finally, you image this stuff in NanoSIMS, and then you correlate the images. It's not like you really take the images and immediately you correlate them. No, because there is always some shift. And the problem is with NanoSIMS, you have very slow imaging. There is always some drift while it's imaging, resulting in a somewhat compressed image. You have to do a bit of morphological stretching and morphological changing of the images to get them to overlap perfectly, but it's not that terrible. I mean, it's not anything that you wouldn't have to do with correlation with EM or with similar techniques. And you can look at all sorts of things. I'll give you a couple of examples and then we'll switch to something more interesting, but you can really look at all sorts of things. I mean, you can look at here at some synaptic vesicle. There was some question of the nature of some active and some inactive vesicles, and then we just correlated them to neurons being fed by 15N. We looked at the 15N to 14N ratio, and we realized, you know, through this that the active vesicles were rich in 15N, you know, were hot, and the inactive vesicles on average tended to be much cooler, much less 15N, implying that the active vesicles are like new, and the inactive ones, the reserve ones called, they are old retired vesicles. So it kind of makes sense, you know, that the synapse in our brains would use new stuff and would kind of like not want to use the very old vesicles. So other things are, you know, you could look, put all sorts of drugs. You know, we put here tetrodotoxin, bicuculin, drugs that change synaptic function and the potential of using vesicles. Did a lot of stainings, correlated a lot of things with the turnover ratio, again, using 15N and 14N. And here, you know, the conclusion, I don't want to waste your time with all sorts of details, but here's the conclusion was that synapses that work hard get new proteins pumped into them. So if you're a synapse and if you're working hard, you receive more new proteins than a synapse that works less hard. So, you know, this again kind of makes sense. The reviewers for this paper said, well, you know, everybody thought it was like that for ages, but nobody's actually shown it. So yeah, I mean, that's basically because it didn't have a tool like NanoSIMS to be able to show something like this. But with SIMS, it's not that difficult to ask such questions. You can look also in the brain of animals. This was a question in the brain of the mouse, trying to look at something like the extracellular matrix, you know, in between cells in the brain of the mouse. and you're looking at some characteristics of it. And again, you have here the extracellular matrix and some other molecules inside the cell labeled, and then you do this 2-color fluorescence and you correlate with your SIMS, and then you get all sorts of measurements. And it turns out, this helped us describe some new type of brain plasticity, which, if you're interested, it's published. I'm not going to go much in detail about this, but it was basically quite nice to be able to look inside the brain of a living animal and just say, how old is this part? How old is this part? How old is this part? Looking where it is, looking how it's organized, and then just asking, well, how old is it? And you couldn't really do this with other tools, as far as I know. Okay, let's now come back to our old questions. So what we asked originally was, what is going on with these doped mice? If you remember still the question, we injected these mice with erythropoietin. They were better at their muscular stuff, but they were also better at their mental stuff. I mean, they were smarter. They could figure out tasks faster than normal mice. So how do they do it? Do they have more neurons? Well, yes, that was basically the answer. People not in my lab, but in the lab of my collaborator, they counted all of the neurons in what is called the hippocampus here, which is smack in the middle of the brain. It's a structure that allows us to coordinate memories, among other things. And you basically had 20% more neurons in the hippocampus. But when they did some labeling with a DNA dye, let's call it like this, to look at which neurons were new in there, were newly born, there was nothing. You had some new cells here, these orange things, this orange dye is here. You have some new cells here and there, but in the relevant parts of the hippocampus, there was no new cell. So how could you have 20% more neurons, but no new cell whatsoever? Really weird. Well, we then looked in NanoSIMS, and we just looked at the levels of 15N divided by 14N. And what you see here is a whole bunch of cells in control, and they are all about the same. In the erythropoietin-injected animals, every once in a while you have one of these cells which is much brighter. Everybody else is the same, but this cell is much brighter. You see it here. It's full of very bright spots inside the cytosol, and the nucleus is somewhat brighter as well. What happened is that this was a small, tiny precursor cell at the time of injecting erythropoietin, and the injection of erythropoietin made this cell grow into a neuron. That's why it doesn't look like it has divided or it has new DNA in there. This DNA dye doesn't pick it up, because this was not a new cell, it was an old one, but it turned into a neuron. It used to be a tiny little precursor cell, then we injected it and it turned into a neuron. So essentially erythropoietin induced the formation of neurons in the brain from old precursor without cell division. All of the tools that are using fluorescence only look for cell division. NanoSIMS could just look at, did this cell really develop? Did this cell really get a lot of new protein in the last few weeks? It certainly did. And it turns out these are about 20% of the cells. So basically erythropoietin made new neurons from old precursors, and this is why these mice became smarter. And we analyzed this in some more detail, later on, and it turned out, and again, I don't want to waste your time with a lot of details, it turns out the immune cells of the brain also changed in behavior, and they became more active to some extent, but they decreased to another extent in numbers. And this kind of allows you to have a nice, cool, not inflamed, normal brain with more neurons, makes you a little bit smarter. I still wouldn't suggest that anybody starts to dope themselves with erythropoietin just to get smarter, because these mice, because they get a bunch of new neurons, may also get epileptic fits, and this might actually shorten their survival. So is it a good idea to use erythropoietin? Probably not. In the end, probably not. So what else can we do? We could correlate with electron microscopy. That also works quite nicely, and you can look here at some electron microscopy of myelin in the brain, and these are various EM pictures, and these are your NanoSIMS pictures. and you can analyze different components of the myelin. And this paper has shown that myelin has different turnover characteristics for different parts. I'm not a myelin expert so much, so I'm but it looks like this thing here is called some sort of a lip of myelin, whatever that is. It's some special part of myelin. And it turns out this type of component has a special turnover. It's different from the rest of the myelin, and as myelin is a very important element in a lot of neuromotor diseases, then this is definitely an interesting tool to search into what is going on with myelin. You could also correlate triply. You could do fluorescence, electron microscopy, and SIMS. You could put your samples in a plastic that allows fluorescence. You cut them, you image in fluorescence, then you image in electron microscopy, then you image in NanoSIMS. Always the NanoSIMS has to be the last because it is damaging. It just burns your specimen away. So then you should always use the NanoSIMS at the end. But you can get a nice triple correlation. And this triple correlation allows you to look at all sorts of weird stuff. So for example, we looked here at electron microscopy showing us morphology of cells. Light microscopy showed us where a specific dye was taken up by active mitochondria. And then we look at NanoSIMS. And we didn't do any pulsing, any feeding with anything. We just looked at NanoSIMS and just said, what are the different ion compositions here? And weird enough, you're seeing here, you have here a couple of mitochondria. They look exactly the same, but one of them is not active. This one here, the other ones are active. And you can actually tell there are some differences, for example, in the nitrogen to carbon ratios in these different mitochondria. The fluorescent ones have more nitrogen to carbon, probably having something to say about the metabolites in there. We didn't really test this into more detail, but it could be done, and you can appreciate. You can get a lot of information, even some really unexpected. What is really there going on type of information. So yeah, not all mitochondria are the same, even in very simple fibroblasts. It could be that these guys, that are not taking up the dye are very old and they have some problems here. Could be, I don't know. We haven't really checked further, but this kind of stuff could be done. So now let's switch gear a little bit and look a bit more into, all of these things are correlative techniques. You correlate to get some sort of idea about what is being labeled. I love the correlation to EM. I think it's the best thing you can do. You get an enormous amount of information, but let's say you don't want to correlate. Not everybody wants to use several instruments for one question. So what about labeling your target specifically in NanoSIMS? We did do some work over that. Initially we made some molecules that carry fluorine, and then you can put them onto your, in this case, it was endoplasmic reticulum, and then you can see the stuff directly labeled in NanoSIMS, which allows you quite a nice correlation to for example, the turnover of proteins with, again, an amino acid carrying N15. You can put the 19F on some sort of antibodies or nanobodies, and then you can just see it in an immunostaining here. These are peroxisomes in these cells, and basically you just immunostain using 19F. So this is quite nice. You can use other things. You can use 15N directly. 15N is even a ratiometric sort of dye. If you use it like a dye, let's say, it's not a great signal to noise, but it's ratiometric, so it might have some advantages in the fact that you can kind of like estimate the amount of molecules you have there because of this ratiometric thing. I must say I don't like it so much because it just doesn't have that great a signal to noise, so that is a bit of an issue. Fluorine is much better. We use boron here, 11 boron, and you can get your nice labeling there with various types. You can put it on antibodies, nanobodies. You can put it in click chemistry. There is lots of ways of doing this. More recently, this is submitted. That's actually the wrong label. It actually came out I think in the summer in Chemical Communications. So this paper actually did come out already. You can use iodine as well. You can use fluorine and iodine. And you can look at a couple of things, couple of different proteins or different identities here, and then you can look at your various things in the cell and ask other questions specifically about these areas. in the end, all of these probes require you to be quite good at chemistry to make them, because none of them are commercial. You can't buy them from anywhere, so you have to make them yourself. And I can tell you that the iodine one was horribly difficult to make. It took us years to get one that really worked. The fluorine is a bit easier to make. Now, of course, now that the protocols exist, you could make it in a matter of weeks. I mean, now it's not so much of a problem. It's just figuring out how to make iodine and boron. I mean, figuring out how to make boron and iodine really usable was an important, difficult thing to do, which Zelda Kabatas in our lab, really mastered. But in the end, it's like, how many people are going to do this? How many people are going to make their own probes? It's not easy. So let's just use some simple commercial probes, see what's going on. So we tested a lot of gold antibodies and say, well, which ones are actually working well? Claude Lechen, who was mentioned in the first lecture, he also used this a few years ago and found some good results with antibodies that he labeled directly in the lab. We said we don't even want to do that. We just want to buy something, put it on, see if it works. And you can actually do it, and it turns out with a medium-sized gold, you get like a very good performance. If the gold is too big, you get a sparse labeling. If the gold is too small, you get a good labeling, but you don't see the gold because it's just too little gold in there. With a medium size, about 6 nanometer gold, you get actually pretty good performance, and you can see your stuff quite well. So that was good. So you can really do a lot of things. You can label stuff, you can study turnover. But what else can you do? I mean, just for the last couple of minutes in my talk, what could you do with NanoSIMS that you could not do with other tools and would be medically cool to do? And the answer is quite a few things. You could look at like the oxidation of cells, like burning of living cells, how they burn in oxygen, and you just image oxygen. Just put an atmosphere of oxygen 18 in there and just see what's going on. One caveat to this, oxygen 18 costs about $1,000 per liter, and a mouse will breathe, as far as I remember, something like a liter per 15 minutes, something like that, maybe even shorter. A mouse will just completely use up 1 liter of oxygen. So that's a tough, expensive experiment. With cells, it's a bit easier to do, and then it's more efficient. Importantly, you could image drugs. And in the last couple of minutes, I want to show you this example. This molecule is called ANLE 138B, and you can purchase this from Sigma Aldrich if you want. What is it good for? Well, it was initially described to block bad oligomers in Parkinson's disease and in prion disease. So Creutzfeldt-Jakob disease and Parkinson's disease, mouse models of this, if you feed them ANLE, or well, actually inject ANLE, they will live longer and be happier. Then it also worked in tauopathies. These are Alzheimer's disease problems. Then it also worked in A-beta pathology, again in Alzheimer's disease. Then it also worked in multiple systems atrophy, another type of aggregopathy, where you get molecules to aggregate and destroy neurons. Finally, it also seems to have implications for cancer. It seems to work and as you see here, has certain effects on melanoma cells. That's interesting. Where does it go? Where does it go? Let's say in the brain. Let's forget about melanoma. Let's just look at the other brain diseases. Where does this go? You can't image it, so nobody really knows. You can't put a fluorophore on it because you see how small this molecule is. If you put a fluorophore on it, it's not really ANLE anymore. It works, God knows what it's going to do, because it's definitely not going to be ANLE anymore. But you did notice it has a bromine. So if it has a bromine, you must be able to image it. And that's what you can do. You can now take brain pieces and just look at where ANLE is. And this is your nitrogen, this is your sulfur, and this is your bromine. It's not even that difficult. Bromine is actually pretty easy to image. The rest is more issues of getting the right questions, the right biological samples, the right tissue. Right now we are looking also at the pancreas with this. I mean, there are certainly lots of things you can use. So if you want to image a small drug, well, there's not that many systems that will do it as well as the NanoSIMS can do it for you. So that was it from my side. I hope I didn't go too fast, but I think we're kind of running out of time. I would like to thank several of our people. New, who is now a professor in Gothenburg, Sinem, who is a group leader in Heidelberg, Ingrid, who is a leader, an industrial project leader in Saarland, and many other people from the lab that some of them recently left. Here, unfortunately, Zelda Kabatas is missing also from the list, who did all of these chemicals, and she's also an industrial leader at the moment. So yeah, that's I would like to thank them, and I would like to thank you for your attention. And I will then unshare because then I could probably see you. How do I unshare? Click this button. I unshare, maybe I can see you, and then hopefully there are some questions.
Yes, thank you, Silvio. Very interesting questions, please. Michael, do you have any in the list?
No new questions, no.
No new questions.
Okay.
Well, I would like to ask you now about this bromine. That's also basically the same question. You have these molecules, and they are, one molecule contains one bromine. So would you have a feeling like how many bromine molecules then we could actually detect?
It's just a matter of signal to noise. You know, it's not a matter of how many molecules you have there, but rather, well, I mean, you know it better than I do. I mean, it depends on how much you can ionize. You can always ionize bromine. It's actually working quite well, so you can always see something. The question is, how much bromine is there how much, let's say, how many bromine readings are you going to get in a native cell? And as bromine is very, very unabundant in a biological sample, then I think you are actually very, very sensitive. So I would imagine your levels are in your concentrations there are probably below picomolar for sure.
Okay, so that's, yeah. So sorry, I had to press some button here.
No
okay,
worries.
thank you very much. So I think, yeah, I really liked all these talks. I think we feel very happy, very lucky, sorry, that we actually chose all these speakers in such a way that they could show us all the breadth where the NanoSIMS can be used in the medical applications. And I would like to thank you again very much for taking the time and for explaining to us what you are doing and how it is all related to medically interesting questions, and also how you mentioned the basic ideas about what NanoSIMS does and how you prepare the samples. That was also very useful. So yes, I would like to appeal to the listeners of this webinar to please contact us if you have been inspired the same way as I was, and we will be all very happy to hear from you, to establish maybe new collaborations and things like this. And of course, we will be in touch with our speakers as well, because this is their expertise and experience, long year experience, will be very useful in the future as well. So I think with this, I would like to finish and finalize this quite nice webinar.